Well, why don't we go ahead and get started? My name is John Bishop. I'm the director of the Institute for Engineering in Medicine. And we'll start off with a brief introduction to the Otto Schmitt Distinguished Lecture Series, and then I'll introduce our speaker for this particular lecture. If I could have the next slide, please. So this is the Otto Schmitt Distinguished Lecture Series. Next slide. So the Otto Schmitt Distinguished Lecture Series is designed to foster discussion of the grand challenges for engineering and medicine. IEM invites national leaders in research, industry, and government to give these talks each year. The lecture is named after Dr. Otto Schmitt, a longtime university professor of biophysics, bioengineering, and electrical engineering, who made significant contributions to a wide range of fields, including submarine detection, electrocardiography, and quality of life measures. He's also credited with helping to found a large number of professional societies, including the Biomedical Engineering Society and the Biophysical Society. And for those of you who don't know IEM, we encourage you to come visit us on our webpage, iem.umn.edu, where we look to turn science and engineering into medical practice. Next slide, please. So this year, it is our honor and pleasure to introduce Gordana Vunjak-Novakovic as our Otto Schmitt lecturer. She is the university professor, the highest academic rank at Columbia University, and the first engineer in the history of Columbia to receive this high distinction. She's also the McCarthy Foundation Professor of Biomedical Engineering and Medical Sciences and a faculty in the Irving Comprehensive Cancer Center, College of Dental Medicine, Center for Human Development, and Mind-Brain Behavior Institute. She directs the Laboratory for Stem Cells and Tissue Engineering, Columbia's Center for Dental and Craniofacial Research, and the NIH Tissue Engineering Resource Center. The focus of her research is on engineering functional human tissues for regenerative medicine and studies of development and disease. She has published 3 books and over 400 journal articles. Her H-index is 124, and she's founded 4 biotech companies with her students. Among her many recognitions, Gordana was elected to the NIBID Council of the NIH, the HHMI Scientific Review Board, the AMB, where she was the chair of the College of Fellows, and was inducted into the Women in Technology International Hall of Fame. She gave the director's lecture at the NIH as the first woman engineer to receive this distinction. She received the Clemson Award in 2009, the Pritzker Award in 2017, the Shu Chen Award in 2018, the Termis Award for Innovation and Commercialization in 2019, and the NCAT's Aspire Design Challenge Award for Translational Innovation in Pain, Opioid Use Disorder, and Overdose. She's a fellow of the Biomedical Engineering Society and the AAAS, and one of the Foreign Policy's 100 Leading Global Thinkers in 2014. She was recently decorated by the Order of Karađorđe Star, Serbia's highest honor. She's an elected member of the New York Academy of Sciences, Academia Europaea, Serbian Academy of Arts and Sciences, the National Academy of Engineering, the National Academy of Medicine, the National Academy of Inventors, and the American Academy of Arts and Sciences. Today, Gordana will be delivering a lecture on engineering human tissues for medical impact. Gordana, it's a pleasure to welcome you to Minnesota virtually for this lecture.
So, John, thank you so much for this wonderful and very, very generous introduction. It is a huge honor to be invited here and to give the Otto Schmitt lecture. I'm very grateful that you actually talked about Otto Schmitt, because I was planning to do that, but you have done it much more nicely than I would. And just being connected to him in any way, even as a lecturer, in this series that actually holds his name is really fantastic for any of us biomedical engineers. So I'll talk today about engineering human tissues for medical impact. And I will try to describe the concept that we follow. How do we do that? How do we know how well are we doing it? And I will mostly try to speak about tissue engineering through examples. So a good starting point is just to remind all of us that the cells really respond and communicate with the entire context of their environment. So when you think about the living cell, it is really responding to the cytokines in the cell environment, to the bioactive molecules, to other cells, to the extracellular matrix, its composition, mechanics, architecture, and to physical forces. And these factors which interact with each other and change in space and time eventually affect the expression of genes in the cell and lead to the outcomes, some of which are very beneficial, such as cell differentiation and self-renewal, and some of which are those that we are really trying to avoid, which is the apoptosis and cell death. These interactions also go in the opposite direction because the cells are secreting cytokines, they affect the neighboring cells, make and break tissue matrix, and generate forces. There is this huge complexity of biological context that we cannot possibly recapitulate using our engineering tools. But I believe that it is incredibly important that we use this knowledge as a guidance when we are designing our tools. And today I will talk a lot about two critically important tools that we develop in bioengineering. One is the scaffolding material or matrix, which is necessary for the cells to attach to and to form a tissue. So this is like a home for tissue formation. And the culture system that we call bioreactor, which is providing the control of environmental conditions. So the way we are approaching the tissue engineering is with this general notion that the real tissue engineer is a living cell. And what we do is to enable this cell through our engineering tools that we use to control the cellular environment. So with this notion, we go in two different directions. In general, as I will show you through examples, we would start from some either biological question or clinical problem or clinical need. And in order to engineer the tissue of interest, we would start from some kind of stem cells. I will only talk about human stem cells because these are the systems that we are mostly interested in. And then we assist the cells through the provision of the scaffolding material and the bioreactor system to make a tissue. The conditions are obviously very tissue specific. The matrix or bioreactor will greatly vary from one biological tissue to another. And if the conditions are right, you may end up with a piece of human tissue that is cardiac or lung or bone. And I will show you some examples of this. And this is the direction that really leads us to regenerative medicine applications. A parallel track, which is a kind of fast track for tissue engineering that started to evolve some 6-7 years ago, is the one that is popularly called organs on a chip. So using the same principles, you are making large numbers of very small sized tissues that you are connecting by vascular perfusion into the physiologically meaningful units. And I will also talk about this. So my first story will be about bone, engineering human bones. And actually this story started literally a month after I moved to Columbia. Warren Greyston, who is today full professor at Johns Hopkins University, was the very first postdoc, new postdoc, joined my new lab. And Warren Gray came to the lab with this passion that he would like to do something in the area of bone tissue engineering that would leave a mark, that would potentially be transformative. So we started to talk about what it could be, and we decided that the toughest test of our tissue engineering technologies would be to try to make an autologous human implant which is anatomically correct. And some of the most complex bones in our body are in the head and face. So we decided to focus on this area. And then we also decided that probably the highest challenge would be related to the engineering of the jawbone, because this is one of the highest loaded bones in our body. So we picked something very ambitious, thinking that if we can do that, we can do also other things. So this is the vision. So you isolate cells, you get images of the defect, you make scaffold, place it into the bioreactor, infuse with cells, and after a couple of weeks, you would grow a piece of living human tissue that would eventually end up in the patient. So this is still a plan. And actually, as I'll show you later, we are just starting now to do the first clinical studies that are testing this technology. At that time, what we have done was to develop a methodology that allows us to make very precisely anatomically shaped graft of this particular bone in the head, to make also the anatomically shaped bioreactor so that this intricate grafts scaffold is really fitting into the bioreactor chamber as hand into the glove, to figure out how to infuse the cells and how to perfuse the system to culture the piece of bone. In order to determine if this technology is really successful, if we can have an effective repair of bone, we also decided to test if these grafts that are grown in our laboratory in New York can travel to the site of implantation. Because as you may imagine, if this technology becomes a clinical reality, you're not going to grow bone all around the country. They will be grown in centralized places and then shipped to where are they needed. From New York to the place in Louisiana that we picked, because we have really good collaborators there and fantastic animal facility, it's in Baton Rouge. And this is about 10 hours door to door from New York to Baton Rouge, because there is no airport there and you need to drive from New Orleans. So essentially the design was that we have done this study in a pig, human-sized pig. This is the animal which is pretty much gold standard for any reconstructions in the head and face. And each of these animals was imaged to get the CT computer tomography data for fabricating the scaffolds in bioreactor chambers. And each animal also donated a small liposuction, the small sample of fat that we use to isolate mesenchymal cells to build the grafts. And then once the grafts were cultured, the grafts and the investigators traveled back to Louisiana. So this is just shown schematically here. So for the envisioned human application that we were testing in this animal model, we would take CT images of, say, fractured jawbone, temporomandibular joint. This is your lower jawbone. And then we would get these three-dimensional files. We would make this scaffold that you see here in my hand, just to get a sense of scale and geometry. We would use the same data to make the bioreactor chamber and insert scaffold into the chamber. And then you infuse the cells, and after 5 weeks of culture, you get a piece of living human bone. So to do that, we had to do a lot of engineering, including the development of this bioreactor system that allowed us very close and precise control of the conditions inside the cultured bone. And we also had to make this a bioreactor portable, because if you remember, I mentioned these grafts needed to travel and stay alive and in a good health from New York to the implantation site. In a separate project, we also managed to place the layer of cartilage over this underlying bone. And the cartilage was made using the same cells and interfaced with bone very nicely, as you can see here on these images. And most importantly, the mechanical properties of cartilage, we were able to bring them into the physiological range, meaning specifically that the young modulus, the compressive modulus that we measured, and the friction coefficient that we measured for this human cartilage were exactly in the range for healthy human cartilage. So this looked promising at this point, everything that we have done in vitro to culture bone and cartilage. So in a more recent study that we just completed last fall, and it's coming out actually tomorrow as a published paper, we have very rigorously testing this technology in a large number of animals. So this was this Yucatan mini pig that I mentioned. So the approach was, as I described to you, autologous cells obtained from the fat samples of the animal that we used to isolate mesenchymal cells and to induce them into chondrogenic and osteogenic lineages, fabrication of the scaffolds for each animal to exactly mimic the particular anatomy, because this anatomy both in people and in animals really varies from one individual to another. The critical component of the project was the development of this dual perfusion bioreactor that I'll show you in a little bit greater detail, but very briefly, cartilage and bone require very significantly different conditions for their formation. So bone requires perfusion and a culture medium that has a specific composition, which is inducive and conducive for the bone formation, while cartilage requires shear forces and chondrogenic medium. And we were able to provide these two different conditions to the grafts. And then the grafts traveled back to Baton Rouge, and we were implanting them, replacing the perfectly healthy, normal jawbone with this engineered graft, and looking how these grafts integrate and remodel and replace the function of the native graft. So this was the project, and actually two key people, two graduate students that were leading this study were David and Josephine. So David is just about graduating and Josephine in a few years. So this was really their study. So this is the bioreactor that I mentioned to you. So there are these two pieces that, like two very soft envelopes that you place around the graft. And at each of these sides, we have like an array of channels. Through these channels, you perfuse culture medium through the bone. And the channels are designed, their diameters are very precisely designed depending on the thickness of the bone, so that a bone is like throughout the bone volume, the cells are all getting the necessary amounts of oxygen and nutrients and being exposed to the necessary levels of shear forces for bone development. And then there was a flow of culture medium, of chondrogenic culture medium over the layer of cartilage. So you can see it here maybe more clearly. So you see here, this is just a computer simulation of the flow of these two different culture media that you see at the right side. I will just play it one more time, horizontally through the bone and on the top over the layer of cartilage. So in this big study, we compared three experimental groups. So the first group that you see in the top row are these cartilage bone complex grafts. So this This is the graft that we really wanted to grow, that would be used to replace the patient's shattered or non-existent jawbone, or jawbone that had to be removed because of cancer surgery, or jawbone that hasn't existed because of congenital abnormality. The control groups were actually the same graft without the cartilage layer. So this is the middle row, to evaluate if cartilage is really critical for the functionality. And we also did some additional studies using very small samples of these grafts to do some detailed biological evaluation. We also studied the biomaterial alone, because you very often get this question, why is not biomaterial sufficient? Do you really need the cells? So to answer all these questions, we evaluated the animals over 6 months. I also want to mention that the choice of scaffolding material, which is a mineralized bone matrix, allowed us to grow bone without the addition of any growth factors, which is really convenient for clinical application. And the other thing is we use methodologies that are normally used in clinic for humans, although this was animal study. And the fixation type that we used allowed us not to immobilize animals. So the pigs were allowed to move around and eat, like start chewing immediately after they woke up from surgery. So what you see here at the top left is actually that scaffold alone resulted in very large area, which is outlined here, which has not remodeled after 6 months of implantation. In contrast, engineered bone developed much better, reconstructed much better than native graft, with maybe some 25% or so of the area of the old graft that has not remodeled. The appearance of the bone that you see here actually looked pretty good, with very nice mineralized matrix, nice soft tissues. And then what you see here, these like small structures are blood vessels that we confirmed here at the right. So we were staining both for endothelial, for vascular cells markers, van Willebrand, and we also stained with Prussian blue, which is a stain for iron, because if you see iron, it means there is hemoglobin. If there is hemoglobin, it means there are blood cells. So the bone looks pretty good. Then in our cartilage bone grafts that you see here at the right side, we were able to recapitulate really nicely this stratified architecture of the native graft. And I'll show you that, show this to you in a little bit more detail. This is the entire condyle. This is the joint. At the left is the bioengineered joint. At the right is the native joint. And you can appreciate that there is a lot of similarity at that time point. And this is a larger magnification of the picture that I just showed you. So you see that in bioengineered graft, you see this multiple stratified layers of cartilage, like from this fibrocartilaginous layer at the top to the middle zone, deep zone, interface with bone, and the actual bone. And then when you compare it to the graft, which is the native tissue at the right, you see that together the bioengineering tools and the in vitro environment followed by the implantation environment have resulted in a joint that has many of the desirable features. So on this picture, I just want to tell you that we have done a lot of cell tracking, trying to understand and confirm that actually it is the implanted cells that were driving the repair. And then you see some of this labeling here that allowed us to track over time how the tissue was developing. And then just to further confirm something that I told you before, so when we compare the compressive modulus of our cartilage bone grafts, which are shown here in the middle, with the native grafts, they were comparable Young's moduli and the bone alone graft had a higher modulus, which is different from what you would see physiologically. And then likewise, the friction coefficient was very similar and low, physiologically low for cartilage bone grafts to the native tissue, unlike bone alone graft. And this could be very nicely correlated with the presence of specific molecules, like lubricin is really important. Its presence in the cartilage is very important for this frictionless behavior, and then aggrecan is very important for the compressive behavior. And this was done, the studies were done actually by using an apparatus that really simulates the joint motion. So we were testing our condyle against a layer of cartilage, which would mimic the contralateral side of the joint in synovial fluid. And then the current situation with translating this technology is that actually some years ago, we launched EpiBone. This is a company that's based on this foundational technology. And then now, after some delays during the COVID, the company is recruiting patients for one of the products from this family. So a clinical study is starting. This is very exciting, and this is terrifying, as you may imagine. So this is now becoming real, and we will know probably in a year from now, year and a half, how effective this technology may be in the patients. So this was my first story out of 3. My second story is about the lung. So lung is the organ system that we never thought about too much. The lab was really focused on bone and heart over the years. And what happened is, this is just a perfect example of commissioned science that is totally driven by the clinical need. So some 7-8 years ago, the transplant surgeons from Columbia visited us to talk about one major problem that they have, and this is they don't have enough lungs for transplant in general. So many patients that are suffering from end-stage lung disease just cannot get the only life-saving therapy that is available. To make this crisis even more serious, they reject the majority of lungs that are received because they either had some injury that was not noticed at the time of harvest, or they just suffered from hypoxia and other factors that are associated with transport. Lungs are very demanding in terms of oxygen levels, and they don't travel very well. So not enough lungs for transplant. Lung is an incredibly complex organ. So what you see here are the images of the actual human airway and actual human lung vasculature. So it's still debated how many cell types are there. It's like between 45 and 50, depending to whom you talk. And there are 24 levels, hierarchical levels. So it's just daunting to think that we may be able to make even a small piece of lung. So between the airway and between the vasculature, you have this gas exchange surface. So it's epithelial cell, endothelial cell, and the basement membrane in between. This gas exchange surface that you have in your chest is about 145 meters square. And just to give you a benchmark, the tennis court is 190. So this is huge. So what we started to think about is, can we do something differently except going and making lungs from scratch, which we don't believe we can do. And actually some attempts to strip the lung of all cells, repopulate with cells, implant in small animal models, show that these lungs were failing very quickly, and it was largely because of the leaky, underdeveloped vasculature. So that was a huge problem. But talking to clinicians from the very beginning gave us some very important information. So we started to work together, and we realized a couple things. One is Maybe we can start from the lung that's not acceptable for transplant and just fix it, improve it sufficiently to make it suitable for transplant rather than making something from scratch. So the first condition was we needed time to do that. So we needed a technology that would keep the lung long enough outside the body, the donor lung, to allow us to improve its structure and functionality. So this led to the development of technology that we call cross circulation, actually connecting the lung to the living recipient. And we have developed this technology by connecting either porcine or human lung to the pig. Again, our same animal model, which is very close metabolically and in many other ways to human. I'll tell you a little bit about this technology. So we bought time by developing a methodology for maintaining lungs under physiological conditions outside the body. The second decision we made is we were told by the surgeons that in many cases the injury is local. It's due to, say, gastric aspiration, like the stomach juice that comes into the lung, or local ischemia or something. So you can find these patchy areas that are in bad shape within the lung that is in a reasonably good shape. So we developed methods that allow us to visualize where are the areas that are in the worst shape and access specifically these areas, removing the defective cellular material and bringing in, as needed, the reparative cells. And then the third piece was also very important. We were continuously told that there is no way to do any lung bioengineering transplant if we don't maintain the integrity of vascular networks. So because the most of the lung injury that we need to fix is on the epithelium, so these are the epithelial cells, they're sitting on the basement membrane, very, very thin collagen laminin membrane, and underneath is endothelium vascular cells. So we found a way to remove epithelial cells while fully preserving the basement membrane and vascular flow, and then replace it with a fresh epithelium. So these three pieces actually translated into what we have done with the lungs. So you see the lung here, which is supported by the animal, which is a pig, which is under anesthesia. The lung is ventilated. It's placed on ventilators that are normally used for patients. And the connection between the lung and the animal is really blood perfusion. Why is this important? Because the animal is really serving as metabolic control. It's providing all this systemic homeostasis and clearance of the factor from the lung. So this is the usual setting in the OR that they have for lung transplant, plus a couple more instruments that we use, such as thermal imaging, for example, and then intravital imaging with some very, very thin bronchoscopes that we use to have insight into what's happening in the lung. So here is just an idea about the gross images, macroscopic images of the lung to show you how much can be achieved using this. So this is the initial lung at the time of receipt that actually has a lot of these white areas. White areas are areas not perfused by blood that we confirmed by these thermal images. So everything that you see black is very cold, low temperature. Everything that's warm because of blood perfusion is a sort of red towards yellow. And you see that over time, over the 36 hours of extracorporeal support, this lung changes visually from lung that's very compromised to lung that looks better and better, as confirmed here with this thermal imaging. Underneath is actually one of the worst lungs that we treated that looks a little bit like liver, that was very much damaged, that was also able to be recovered to great extent using this kind of support. And just to show you a glimpse into functional data, what you see here at the left are so-called pressure volume loops. So your healthy lung is supposed to look like this here, like the control in the far left. So you don't need much pressure to achieve the volume that you need to inhale and exhale. The lung that we received, the lung that was top left in the previous slide, is this red lung. So very low volume and really transported into and from the lung with much higher pressures, about twice as high as normal. During the recovery, we came from red to this light blue here. You see, this is not normal, but actually it's close to normal, and this is what the transplant surgeons would declare as acceptable. If you look at this graph here, you see that actually this very complicated ratio is essentially the efficiency of oxygen utilization, how much oxygen that you inhale into your lung gets into your blood. And we started low, and then we came, this dotted line is the physiological level, so you came to physiological level. So the cells are smart, they can really recover the tissue in many cases, including this one, if you provide them with the right conditions. This is a development from earlier last year and early this year. So for some lungs, you need more than these 36 hours that I showed you. And we had to develop a system that allows a living animal that's like moving around and playing and eating and whatnot, supporting the lung for a longer period of time, and we came to 100 hours. This is what was our protocol that we were testing, and this allowed us to actually recover some lungs that were not recoverable over shorter periods of time. It's also very important to understand we never aimed, or we will never be able to recover any lung. It is just the idea that we would really like to recover some of them and increase the number of lungs available for transplant, because today transplant surgeons reject as high as 80% of the lungs that they may get. And if you save like this 2 out of these 4 lungs that are rejected, you would triple the number of lungs that are available to patients. Another option that we are considering is, the idea is that the patient who is already on extracorporeal support may potentially, in some cases, support their own lung during the recovery. This is just another clinical option. So most recently, this study that was finished and published this year, we were able to translate this technology from porcine to human lungs. And I just want to show you 2 slides. One is this one. What you see at the left is actually the lung that was a human lung that was rejected by 3 transplant centers, each of them testing it and trying to recover it, and then it ended up as a research lung. These are the lungs that we can obtain that are clinically not useful. And we were able to actually recover this lung to significant extent, both by appearance and structurally and functionally. And maybe the best way to see it is this one. This is the initial lung that we received. And then it's just a time lapse. So you will see the initial picture. This is after 12 hours, and this is after 24 hours. So this is the human lung going through this treatment and support on cross circulation. I mean, this got some traction, like this result. And I think what we were mostly excited is actually enthusiasm of the surgeons that are continuing now to work with us to see how to manage the clinical translation of this technology, which is a really non-trivial task, but we are very, very enthusiastic in trying to move this to the patients. And we are also trying to use the same methodologies. This is just the very beginning of the whole effort for this targeted removal of the cells, replacement of the cells, targeted treatment in situ, which should be possible in principle. These studies are again done in a pig model, and they're also done in the context of COVID, which is one of the like the most significant consequences of COVID is so-called ARDS, acute respiratory distress syndrome, that we are trying to treat in a porcine model, developing a new type of therapy and seeing if this can help people who have ARDS for all sorts of reasons, including this horrible infection that is really turning upside down our lives these days. And let me just now switch to the third and last piece that I would like to present to you. I mentioned at the beginning that in parallel to regenerative medicine modalities, two of which I try to illustrate to you, there is also an effort to build better models for studies of disease and testing of new therapeutic modalities. This is an effort that was initiated by NIH about 7 years ago. And the motivation is that the drug development in principle is one of the most expensive and least efficient technologies that we have today. This is a typical scenario. So the drug discovery starts from very large number of compounds. There is a huge attrition when you go through preclinical and clinical trials and even post FDA approval history of the drug. And this is largely due to the current systems, which is a simple cell culture and animal models, mostly rodents, not being representative of human condition. So the NIH asked essentially if advances in stem cell biology and bioengineering, tissue engineering, could be combined into building human tissue models that would do a better job in avoiding false positives and false negatives and de-risk the drug translation into clinical studies and eventually into the patients. So they actually posed some requirements that we complemented by some requirements of our own. So we tried to make, and this is actually a project, a consortium that has a number of laboratories across the US. So we are trying to make platforms that are configurable and modular. So in an ideal world, you would combine the tissue modules in a way you need them for studying specific questions. The non-negotiable requirement by NIH that was driven by pharma and FDA was that we need to have stable tissue phenotypes for long times. 4 weeks is a minimum, going to months, because this is a necessary condition to do meaningful pharmacological and biological testing. A requirement that we introduced, and I'll show you a little bit about it, is that in order to really maintain stable tissue phenotypes for a long time, while connecting tissues to each other, we separated the vascular from tissue compartment by endothelial barrier so that we can really allow the tissues to communicate to each other like in our body while being maintained in their own, each in their own niche. The fourth requirement was a sort of easy one, just to develop as many functional readouts online as possible, because this allows you to just monitor the status of the cells in your tissues and to do dynamic studies. You just do a longitudinal study without sampling the tissue itself, and this is bringing a lot of new value to your experiments. And then finally, we decided to do patient-specific studies, which means that we work with iPS cells that are derived from blood so that you can have a clean genetics and you can study, like parse out the genetic from systemic factors in the patient. So this is how it works. So we would start from the blood sample. have the iPS cells derived, make these individual tissues that we connect to each other into this kind of a platform. So this is how it looks. So you see like the top view, different types of tissues, the bottom view, you just see the interface with the vasculature. And then on the right, you see that actually how we are assembling this platform and putting it together. This is just an illustration of this requirement that I mentioned to you, that we are making these tissues patient-specific. So it took us 3 years to really develop the methodologies that are indicated here by adopting and adapting a number of different methods. So the iPS cells are really induced to differentiate into component cells of all these different tissues that we are interested in. And as you know, each tissue, with maybe exception of cartilage, consists of multiple cell types. So this was a huge effort to make this library of cells and methodologies to make different tissue types. So this is what we have so far to be used in our platforms. So we have sensory neurons, we have the heart muscle, we have a bone tissue with bone marrow inside the tissue, liver, endothelium, and vasculature. We have tumor tissues, skin. We have skeletal muscle that we study coupled with motor neurons, because we are very interested in neuromuscular junction, and we have the immune cells. So this is the repertoire. You don't use all these tissues in all platforms. You always pick the simplest possible combination that gives you enough physiologic relevance to really emulate some of the aspects of the whole human physiology. This is an example of the heart. So you see this heart muscle beating in response to electrical signals. It has electrophysiological properties that are mimicking some aspects of the native heart. This is an action potential with this very important notch. And you can see that the shape and amplitude of this potential look right. This is just the transmission electron microscopy, where you can appreciate these registers of sarcomeres. This is actually the active part of the cell that is at the basis for these contractions. And the blobs that you see here are mitochondria. So when you see a lot of mitochondria, in this case, it was a physiological level, then you know that these cells are very metabolically active. And this is a cross-section through the muscle where you see the body of the cell surrounded by the cellular membrane. And what is really important here are these little thin lines. So these are these invaginations of the membrane into the cell body that are called transverse tubules, that are instrumental for facilitating the transport of calcium, which is the basis of the heart cell function. So the other example that I can show you is this model of the neuromuscular junction. So this is the work of Alaya Villa, and what she has done was to start from the same cells to derive myoblasts and to derive motor neurons. And what she has done is that the batch of the cells that she used to derive motor neurons, she edited optogenetically to make these cells light sensitive. So this was, she actually intervened on this channel, rhodopsin channel. So basically when you shine light, then this neuron is firing a signal. And the myoblasts, the muscle cells that are derived from the same source, are not sensitive to the light. So this was step number one. Step number two was that she made out of motor neuron precursors, she made this neurosphere that you see here, like a little ball of cells. And she placed it into a microfluidic device that you see here at the left with channels that were guiding the extensions of axons downwards. And what you see at the bottom of this image is this red structure shown schematically is the skeletal muscle that was built the same way we have built this cardiac muscle that I showed you like 2 slides ago. And you can see it here more clearly. So this is shining green, and I hope you can also see these green shining axons. So as you shine light of the appropriate wavelength onto this construct, then neuron should be firing. And if the connection is functional, the muscle should be twitching. I'm not sure if you really see twitching here, but maybe that will be easier on this other. Let me just show you another image. So at the left, you see how it looks. So muscle is a sort of red orangey, and then the neurons are green, and they're like physically connecting each other. But then to show that there is really coupling, you can look here at this normal muscle, and what you see here is a signal and the response, and the score that is labeled here of 78% means that 78% of stimuli has a response. So this muscle is responding pretty well. Then we added into this exact same culture of healthy muscle coupled to motor neuron serum that we obtained from a patient with myasthenia gravis. And this is a disease that is described by dysfunctionality of the neuromuscular junction. The presence of this serum actually inhibited any response of the muscle to the stimuli. And at the right, you see some hard data that were obtained actually in a very automated, this was one of these algorithms that we developed to have an unbiased evaluation. So you see that the presence of myasthenia gravis serum, which will be like this bar in the middle, results in 0 readouts. And then when you rinse away this serum, then the muscle recovers and starts beating. And we are doing like lots of research in collaboration with many of our colleagues on these platforms. We recently started to work on a cancer patient on a chip. This is a new project in our lab where we are modeling the cancer metastasis, selective targeted cancer metastasis into bone tissue, liver tissue, and lung tissue. We are also giving these platforms away to our colleagues in neuroscience and those studying diabetes. So we are really trying to make the best use we can out of them. One of the recent studies is just schematically shown here. You can do a lot with something very simple, which is just a combination of a tumor that was grown in bone, one of the sarcomas, and heart muscle connected by perfusion, as I showed you. And the rationale for this was that we wanted to recapitulate the clinical trial of a new anti-cancer drug called Linsinetib that was tested in Europe, and where the clinical data did not recapitulate data obtained in preclinical models. So we wanted to measure the efficacy of the drug in treating the sarcoma, so suppressing the cell growth, and also the off-target effects of this anti-cancer drug on the heart muscle. And very interestingly, when we looked into studying these tissues individually, we obtained the result that was a kind of similar to what was obtained with classical preclinical model, with the rodent models. So if you don't have a drug, you just supply your tumor with an empty vehicle, the cells grow. 3 days, 7 days, 21 days, they happily grow in the tumor. When you add the drug, they stop growing, you arrest the growth. But this is not what was seen clinically. When we connected them, then we saw that actually the cancer cells did not really respond to Linsinetib therapy, this particular metastatic cancer had no response, and this is exactly what was seen clinically. So this is just sort of still anecdotal. This is one single study, but we are really trying to work on this in a much more systematic fashion and build with our colleagues from systems biology department, clinicians, oncologists, and others to try to continue with this lines of work. So this brings us like to the last 2 minutes in my last few slides. So to try to summarize, in general, I try to convey the message that we design the environment so that the cells can do their job, the cells can do the engineering, the cells can build tissues. We apply this principle in the area of regenerative medicine, and I showed you 2 examples of very recent studies of whole organ engineering. One is bone and one is lung. Both of these organs, as you know, have phenomenal ability to regenerate if they are healthy. So this is where the bar is probably lower than for some other tissues. But still, when you have a significant injury, then you do need some assistance to the cells in these organs to rebuild the tissue. And they also showed you the example of organs on a chip, which allow us modeling of human physiology in vitro. You can imagine running clinical studies on a chip like we attempted with lensinetab. And one of the, I think, big goals for the entire field is to use these models in conjunction with everything else that we have available to detect the early biomarkers of disease, because this is something that would definitely have the highest impact on both the diagnostics and treatment of disease. And because of the nature of this talk, I think that it's appropriate to maybe mention a couple of challenges and future needs. I think that one very important thing, and this is something that was really nice to hear this morning when we discussed the Institute for Engineering in Medicine, that the best technologies, the most impactful technologies are those that are developed driven by clinical needs. Not the other way around, not developing technology in isolation and then contacting clinicians to see if they can use it somehow. I think that the progress in this area is very largely driven by our increasing understanding of biology. Our designs, our engineering designs absolutely need to be inspired by biology. The more we know, the better job we do. The better job we do, the better we can study biology and maybe learn more about some of the underlying principles. On the other hand, engineering is really enabling biology by providing not only tissues and tools for medicine, but also providing models of diseases, models of development, models of tissue regeneration that we can use to conduct some controlled studies. I think we are moving more and more into patient-tailored approaches. It's not statistical approach, it's not population approach, it's an approach both for whole organ engineering and for tissues on a chip that is really focused on a patient, because we are very different from each other. You know that, for example, cancer is a good example. They are responders and non-responders, and it is in many cases very difficult to understand what makes a patient non-responder. Sometimes patients with much more advanced stage of cancer respond beautifully to some of the therapies and the other way around. So more knowledge is needed through these individualized approaches that can help us move the needle from non-responding to responding patients. And then finally, I think that as you have also done in your institute, I think the whole field is also moving into new modalities for training. The field is changing and our approach to mentoring and training our graduate students, undergraduate students, our postdocs and clinical fellows are also changing. So with this, I would really like to close with thanking my lab. So this is from happy times when we were still working together, you know, in lab in person. I mentioned that we also made a significant effort in commercializing technology. So with the students that you see, we collectively launched a couple companies, and it's very nice that actually all of them are populated in part by the lab alumni. So like the transition of technology really goes from research to commercial effort. A big thank you to our funding sources that keep us afloat. And I give here a list of collaborators because it's very, very important to say that this is an area that totally depends on organic collaborations between people from different disciplines. This is the only way how we can advance science. I think the main progress really happens at the boundaries of disciplines rather than in any particular area. And like a more contemporary image, so this is one of our Zoom meetings. So we all live in Zoom Live these days. And I mean, fortunately you see some smiling faces, and then like we have this like a very different scene in our lab meetings than we had before. So at the end, I just want to thank you all for being here and to thank you for the huge honor of inviting me to give this, this lecture.
Great. Thank you so much, Gordana. That was a really impressive suite of examples in tissue engineering and across both hard and soft tissues. Very inspiring, I think, also with the commercialization aspects and what you've been doing in your training. So I'd like to open it up to questions, and I do see a few in the chat already, so I will just read those out for you. The first one is your success with bone bioreactors is outstanding, and I think I know the answer to this, but are your bioreactors for sale so that others could test different cell types?
Yeah, they're not for sale. I mean, they may be open for collaborations. I mean, I told you like this bioreactor that I showed to you, it will just be published tomorrow. It's not even out yet, but we are always willing and happy to talk to people about sharing knowledge and tools. So just call me and we can see if we can do something to help you in your efforts.
Okay, great. Another question is from the bone technology. Can this be applied to knee and hip bones and cartilage?
Yeah, in principle. So we started from the head. Actually, we have grown some pieces of bone and cartilage that are in the skeleton, not in the head and face. So there are two important differences, as you know, just developmentally. The tissues in the head and face come from the different embryonic layer. It's a different origin than the skeletal. So for example, cartilage in the TMJ, in the jawbone, is a fibrous cartilage, and cartilage in the knee and elbow, like in hip, is hyaline cartilage. So there are differences, but the principle that we used, which is to provide each of the two compartments, the cartilage compartment and bone compartment, with a separate set of distinctly different conditions, can be and is being translated to other joints, to other cartilage bone constructs.
And then I see, congratulations, I presume that's in your native language, from Dan Glumak. And I'm afraid I'm gonna probably not say it correct, but Odincio Presensagi Puno Havla, Dane. I don't know if that means anything to you.
Oh, okay, now I see it. Now I see it. Just a compliment. Okay. Excellent presentation, thanks a lot. So yeah, sorry, I saw his other, I was reading the the other comment where he said that Otto Schmitt was also the inventor of the Schmitt trigger. Circuit can be used to digitize data and signals, very famous in the EE realm, which is true. So
that is true. Yes, thank you, Daniel, for that comment. We'll be sure to point that out in the next Otto Schmitt. We have a couple of more. So this is actually from one of my students, Zonghu. Are the engineered tissues at the same age as the patient? And I'm not sure exactly what he means from that, but maybe you could just talk about the.
No, this is actually a really nice question. There are two answers to this. Everything we derive from iPS cells, from the stem cells, is initially very immature. And the big effort in the field is to make muscle which is more mature, or bone that is more mature, like closer to postnatal, you know, to really, to bring it to the same age with the patient. And the success is very partial. So with some tissues, especially mechanically, electromechanically active tissues, we are doing a better job in maturing these tissues than with the other that are more like metabolic tissues. So this is the first part of answer. This is important. We are working on it. We are only partially there. The other interesting aspect of this question is, maybe this is what you meant also, we are thinking a lot with these living grafts, for example, living bone grafts, if they can find utility in pediatric applications. Because this is an area that absolutely cannot rely on prosthetic devices that stay fixed, you know, like the child grows. And given the very large extent of remodeling of these grafts following implantation and very nice integration, we wonder if like implanting grafts into young individual could serve them for life, like spare them from going through this like a serial surgeries. So I'm not sure like if I have not answered your question, just email to me and then I will try to respond in a way you expected me to do. So
thank you, Gordana. Yeah, another question about the. I think this is related to how much clinical translation or need there is for the bone bioreactors now, whether they become functional quickly enough for clinical applications now, or is this still in you know?
Yeah, this is another really good question because at this point, we need 5 weeks to grow a graft that I showed you, which is also not 100% complete, but it is complete enough for load bearing. I mentioned that the animals were not immobilized. They were able to do normal mastication, normal chewing. So 5 weeks is not such a long time if someone can wait, but it is too long for any emergency procedures. So we are also thinking in the direction that like for patients that may not be able to wait, can we develop a more of like a point of care application where you can more quickly, ideally in the operating room, assemble, so you can make your scaffolding material, can you infuse it with the cells and maybe just very quickly condition and implant. This is completely unexplored territory. So we need to see, it's just an idea, because whenever you talk with clinicians, you get 2 answers. One is, this is really great because most of the patients have a chronic situation of non-functional joints, and they would rather wait for these 5 weeks or so to get a biological graft than to be given prosthetic device. But the others will ask you, can you do something about trauma, you know, and like immediate emergency situation that may not be perfect, but maybe good enough. You know, you place something into the body and let the body finish the job and complete the remodeling. So
Yeah.
it's just an idea. It's not even like we haven't done any work in this direction, but we are thinking about it.
Well, and maybe there's a role also for preservation to help you in the future, Gordana. Right?
Exactly. This is another, this is another, like, can be, we are also thinking, you reminded me on something else. We are thinking, like, this whole technology was developed to show feasibility of growing your own graft. So like the patient's own cells, which is a longer process, and it's also, logistics is more complicated because for every single graft, you need to go through quality control. But maybe we can think of matching grafts, in which case you can have, you may rely on preservation and having the graft ready for a person when this graft is needed.
Yeah.
This is like another whole other option that may become available.
Maybe a conversation to continue later.
Sure.
We have a couple more questions. So how significant were immune responses when the human lungs were connected to the pigs?
Yeah, this is also, like, these are such good questions. So we didn't know initially what will happen. And we just started by default by applying the immunosuppression regime exactly in the way it is used clinically when the patient is being transplanted a donor lung. So this is allogeneic, as they say, like from one individual to another within the same species. And so when you have human lungs supported by pig, this is much more complicated situation. This is xeno because it's different species. But on a short term, which is what we have done so far, we were able to get around with the same immunosuppression regime.
Okay.
However, we just started working with lung immunologists because there is so much to immune response. There is so much detail that we still haven't looked into. And there are also a lot of longer term effects that we would like to look into. So at this point, we are very aware that this is very significant question. And I think this will be the key question that will determine if and how we can translate into the clinic. And we are really working on it.
Just a quick comment on that note. We have a company called Recombinetics in the cities where we humanize pigs, and I know there are other companies. One of the people in the audience today was Perry Hackett, who was one of the founders of that company. So maybe there's an opportunity there as well.
No, I would love, I mean, we have used some of these special pigs from another source, but actually this was, maybe we can just discuss offline. One limitation was just completely non-scientific, that these pigs are very expensive. And so we still have them in our project plan, but we are sort of saving them from the very end to just confirm everything because it's very hard to do, experiments with 20 animals or so at the cost that they involve. But this is something that we are and I would be very interested in speaking with your colleagues, so maybe we can do something.
And I see one more question actually from Chris, who you met earlier. Do you have plans to knock out HLA class 1 genes to avoid tissue rejection? And I think this is related to the humanized pig.
Yes, exactly, Chris. This is part of the plan. This is something that we already discussed. And this is something that the field is, direction that the field is really pursuing, something very, very important. Yes.
So I want to thank you for this lovely presentation and this very, wonderful discussion. And I also want to encourage everybody in the audience, if you didn't get your question answered, please send it to IEM@umn.edu, and I will forward it to Gordana, and I'm sure she'd be happy to.
Yeah, and you can also feel free to email to me directly. I'll be happy to respond and get in touch about any of this.
So I will I want to thank you again. I don't know how to put on my virtual clapping, Gordana, so I'm just gonna say thank you very much.
Thank you.
And it's been a delight to spend a little time with you virtually, and I'm sure the audience feels the same. This was a very special election. Thank you so much.
I'm very grateful. Thank you.
All right, and we'll let you move on to your next meeting, which I think is with the stem cell group.
Yes. Yes.
Okay. Fantastic. Thank you, Gordana. Thank you, everybody, for participating. Thank you to the staff for the excellent help with the AV. Bye-bye.
Bye.
We'll be in touch.